Hey, listener. Ready to dive into a world where bacteria tremble? We're tackling pharmaceutical biotechnology today,
It's going to be a deep dive.
especially for those of you prepping for your BSc Biotechnology Semester VI exams.
You bet. Specifically, we're looking at your question bank special session.
You guys provided us with a seriously hefty question bank.
But hey, the more questions, the better prepared you'll be, right?
Absolutely. Okay, so let's jump right in. First question is pretty broad. Explain antimicrobial agents, types, and examples.
hmm
I feel like we've all heard of like antibiotics, but
Yeah.
where do they fit into the bigger picture?
Right. Well, antibiotics are just one type of antimicrobial agent. Think of antimicrobial agents as like our arsenal.
Eh.
Against all those microscopic bad guys, you've got bacteria, fungi, viruses, protozoa, even parasitic worms.
Wow, so it's like a whole military operation going on at the microscopic level.
Exactly. And just like a real war, you have to choose the right weapon. So you've got your antibacterial agents or antibiotics. Those are the big guns when it comes to fighting bacterial infections.
Okay, so antibiotics are the big guns.
Uh-huh.
Got it. So then what else is there?
Well, you've also got your antifungal agents for fungi, antiviral agents for viruses, antiprotozoal agents for protozoa, and antihelminthic agents for those pesky worms.
So it's like a whole spectrum of defenses, each one targeting a specific type of enemy.
Exactly.
Okay.
And within each category, you have different classes of drugs. And each one works in its own special way, its own way of taking down the enemy.
Okay, so that's where it gets really interesting. Let's start with the antibacterial agents, the antibiotics. What are the different ways they actually attack bacteria?
So bacteria have a few key vulnerabilities, right?
Yeah.
We can totally export those. One is their cell wall.
The cell wall.
Yeah, it's like their fortress, protecting them from the outside world. So some antibiotics, like the famous penicillins, they work by inhibiting cell wall synthesis.
No, they're like saboteurs, weakening the walls of the bacterial fortress.
Exactly. They interfere with the enzymes that bacteria use to build and maintain their cell walls, making them weak and likely to burst open.
Wow, talk about bringing down the house.
Yes.
So what other strategies do antibiotics use?
Another crucial target is protein synthesis. Bacteria need proteins to function, just like all living things, right?
Right.
So some antibiotics, like tetracyclines and macrolides, they actually bind to bacterial ribosomes.
Okay.
Those are the protein factories inside cells, and they mess up the whole protein production process.
So it's like jamming the assembly line, halting production.
Precisely. Without proteins, the bacteria can't survive.
Makes sense. Okay, so we've got cell wall inhibitors and protein synthesis inhibitors. What other tricks do antibiotics have?
Well, some antibiotics, they go right for the source, They target bacterial DNA.
Their DNA.
Uh-huh.
Drugs like quinolones, they interfere with these enzymes called DNA gyrase and topoisomerase Phi.
Okay.
And these enzymes are crucial for DNA replication and repair. So it's like scrambling their genetic code so they can't make copies of themselves.
Precisely. Without these enzymes, their DNA gets all messed up, and they eventually die.
That's pretty clever.
Yeah.
Are there any other major mechanisms we should know about?
Yes, there are some antibiotics that disrupt the bacterial metabolic pathways,
Metabolic pathways.
like their metabolism. For example, sulfonamides, they target the synthesis of folic acid.
folic acid.
Uh-huh.
Wow.
And that's an essential nutrient that bacteria need to survive.
So it's like cutting off their food supply, essentially starving them out.
Exactly. Sulfonamides, they mimic this molecule called PABIA, which is essential for bacteria to make folic acid. They basically trick the bacterial enzyme responsible for making folic acid and block the whole process.
It's like a Trojan horse, disrupting their essential processes from the inside.
Precisely. And finally, some antibiotics directly attack the bacterial cell membrane, their outer layer.
Okay,
This approach is often a last resort for really resistant bacteria.
gotcha. So it's like punching holes in their armor, causing them to leak and die.
Exactly. Polymyxins are a good example of this. They disrupt the cell membrane and cause bacterial death.
Okay, so we've covered 5 major mechanisms: cell wall synthesis inhibition, protein synthesis inhibition, nucleic acid synthesis inhibition, metabolic pathway disruption, and cell membrane disruption. That's quite a toolbox.
It is indeed. And remember, each mechanism has specific examples of drugs, like penicillin and cephalosporins inhibit cell wall synthesis, while tetracyclines and macrolides inhibit protein synthesis.
Okay, so it's not just about knowing the mechanisms, it's about connecting them to specific drug classes.
Right.
This is making so much more sense now.
Yeah.
So what about the other antimicrobial agents, the ones that target fungi, viruses, protozoa, and worms? How do they work?
Well, each of those categories has its own set of challenges.
Yep.
Fungi, for example, they're eukaryotic organisms, just like us.
Oh, so that makes it trickier to target them without harming our own cells.
Exactly. Antifungal agents often target components of the fungal cell membrane, like ergosterol, or they interfere with cell wall synthesis.
So we have to find those subtle differences between fungal cells and our own cells.
Precisely. For example, you've got polyenes like amphotericin B. They bind to ergosterol and fungal cell membranes, disrupting their structure. Then you've got azoles like fluconazole. They inhibit the production of ergosterol, weakening the cell membrane.
Okay, so similar concept to targeting bacterial cell walls, but with a different target.
Exactly. Now, viruses, they're a whole different story. They're not even cells. They're basically just genetic material in a protein coat.
So how do you even target something that's not a cell?
Well, viruses need to hijack host cells to replicate. So antiviral agents target different stages of the viral replication cycle.
Okay, so interrupting their invasion plan at different points.
Exactly. Some antiviral agents, like amantadine and rimantadine, they actually block viral entry into the host cell. Others, like acyclovir, interfere with viral DNA replication. And some, like the protease inhibitors used for HIV, they block the processing of viral proteins, so no new virus particles can be made.
So it's like a multi-pronged defense, targeting their weak points at every stage.
Now, protozoa, they're single-celled eukaryotic organisms.
Okay.
Antiprotozoal agents, they often target specific metabolic pathways or organelles within the protozoan.
So finding those unique features of protozoa we can exploit.
Precisely. For example, metronidazole, it damages the DNA of certain protozoa.
Right.
Then there's the parasitic worms.
Oh, yeah, those.
Antihelminthic drugs often target the neuromuscular system of the worm, causing paralysis.
So it's like a biological weapon that specifically disables their movement.
Exactly. Like praziquantel causes muscle spasms in the worm, and the body expels it.
Wow, we've gone on a world tour of antimicrobial agents.
And for your exam, remember, it's not just about knowing the names of these drugs, but understanding how they work, their spectrum of activity, and their clinical applications.
Okay, that's a lot to remember, but don't worry, listener, we'll break it down step by step. Now let's dive deeper into those bacterial assassins, the antibiotics. I'm ready for the next question.
Hit me with it.
All right, this one's a bit more specific. Explain the different mechanisms by which antibiotics work. I feel like we've already touched upon this, but let's really get into the nitty-gritty.
Let's do it. Starting with cell wall synthesis inhibition. Remember, the bacterial cell wall is like their armor. It's made of peptidoglycan, a special polymer that gives it strength.
Like their brick wall, keeping them safe and sound.
Precisely. So antibiotics that inhibit cell wall synthesis, they mess with the enzymes that make peptidoglycan. They disrupt the construction process, leaving the bacteria vulnerable.
So like sabotaging their bricklaying process.
Exactly. Now, one of the most famous examples is the beta-lactam antibiotics, like penicillin and cephalosporins.
Ah, penicillin, the antibiotic wonder that changed the world.
Exactly. Beta-lactams bind to these things called penicillin-binding proteins, PBPs, enzymes involved in the final stages of peptidoglycan synthesis.
So they're like those inspectors that shut down a construction site because of violations.
Perfect analogy. By binding to PBPs, beta-lactams prevent the cross-linking of peptidoglycan strands, weakening the cell wall and causing the bacteria to lyse. That means to burst open.
So the bacteria actually explode.
Wow, it's quite dramatic. Now, there are tons of different beta-lactams, each with its own spectrum of activity and clinical uses.
So different beta-lactams are used for different infections.
Exactly. Like penicillin G is mainly active against gram-positive bacteria, while ampicillin has a broader spectrum covering both gram-positive and some gram-negative bacteria.
Okay, so it's like choosing the right tool for the job.
Now, besides beta-lactams, there are other antibiotics that inhibit cell wall synthesis. For example, vancomycin. It binds to peptidoglycan precursors, preventing them from being incorporated into the cell wall.
So it's like blocking the delivery of building materials to the construction site.
Exactly. Vancomycin is often used for serious infections caused by gram-positive bacteria, especially those resistant to beta-lactams.
Okay, so it's a valuable weapon for those tougher battles. Now let's move on to protein synthesis inhibition. How do antibiotics actually disrupt the protein-making machinery of bacteria?
Well, remember, bacterial ribosomes are different from human ribosomes.
Okay.
That means we can target them specifically.
So it's like having a secret code that only the enemy understands.
Exactly. Now, different antibiotics target different parts of the ribosome. For example, tetracyclines bind to the 30S ribosomal subunit, while macrolides bind to the 50S ribosomal subunit.
Okay, remind me again, what are those subunits?
So think of the ribosome as a machine with two main parts, the 30S and 50S subunits. They work together to read the genetic code from messenger RNA, mRNA, and translate it into proteins.
So they're like the decoding and assembly departments of the protein factory.
Great way to put it. Tetracyclines, by binding to the 30S subunit, they block the attachment of transfer RNA, tRNA, which carries amino acids to the ribosome.
So it's like preventing the delivery of raw materials to the assembly line.
Precisely. Without those amino acids, no proteins can be made.
Okay. What about macrolides? How do they interfere?
Macrolides bind to the 50S subunit and prevent the ribosome from moving along the mRNA molecule.
So it's like putting a roadblock on the assembly line.
Exactly. This blockage stops the ribosome from adding amino acids to the growing protein chain, effectively halting protein synthesis.
Okay, so both tetracyclines and macrolides disrupt protein synthesis, but in slightly different ways.
Now, another class of protein synthesis inhibitors is the aminoglycosides, like gentamicin and tobramycin.
Okay.
They also bind to the 30S subunit, but they cause the ribosome to misread the genetic code.
It's like introducing typos into the instructions.
Precisely. This leads to the production of non-functional proteins, disrupting the bacteria's function.
Okay, so it's another way of sabotaging protein synthesis, but with a different twist.
Yeah.
Now, I'm really intrigued by those antibiotics that target bacterial DNA, the nucleic acid synthesis inhibitors.
Right.
Can you tell me more about how they work?
Of course. Remember those enzymes? DNA gyrase and topoisomerase II?
Right. They're like the master organizers of bacterial DNA.
Precisely. Quinolones, they inhibit these enzymes.
So they're messing with the DNA management system.
Exactly. They bind to the enzyme and the DNA itself, forming a stable complex.
Okay, so it's like a three-way handshake between the drug, the enzyme, and the DNA.
I like that. That complex formation is crucial. It stops the enzyme from doing its job.
So the enzymes are trapped and can't manage the DNA properly.
Exactly. Normally, these enzymes make temporary breaks in the DNA strands to manage the coiling and uncoiling process.
Right, it's like a delicate dance of cutting, moving, and rejoining DNA strands.
Precisely. But when quinolones are bound, the enzymes can cut the DNA but can't reseal the breaks.
So the scissors get stuck in the cut position.
Exactly. This leads to double-strand DNA breaks, which are super toxic to bacteria.
Right, it's like cutting the DNA's backbone, disrupting its integrity.
Now, the bacteria try to repair these breaks, but the quinolone enzyme DNA complex also interferes with the repair mechanisms.
So it's like sabotaging the repair crew, a double whammy.
Precisely. This leads to bacterial cell death. Now, importantly, quinolones are selective for bacteria.
How do they avoid harming our own cells?
They have a higher affinity for bacterial DNA gyrase and topoisomerase lovey compared to the human versions of those enzymes.
So they preferentially bind to the bacterial enzymes.
Precisely. It's all about those subtle structural differences.
Wow. It's remarkable how these tiny molecular differences can be exploited to create such targeted therapy.
No, we have to talk about bacterial resistance.
Right, bacterial resistance. It's like that supervillain that never goes away.
Unfortunately, you're right. Overuse and misuse of antibiotics are major contributors to resistance.
Okay, so it's like a constant arms race, trying to stay one step ahead.
Exactly. It's crucial to use antibiotics responsibly and only when necessary.
Well, that was fascinating. I feel like I could teach a class on this stuff now.
Uh-huh. Well, hold on. We still got a lot more to cover.
I'm ready for it.
All right, let's move on to sulfonamides. The question is, explain the mechanism of action of sulfonamides.
Yeah, those undercover agents. I'm ready to hear more about how they operate.
So, bacteria need folic acid to survive.
Right, it's crucial for them, and sulfonamides mess with that process somehow.
Exactly. They target an enzyme called dihydropteroate synthase, or DHPS.
DHPS, that's a mouthful. So it's like a key player in their folic acid production.
Precisely. And here's the sneaky part. Sulfonamides look a lot like PABA, a molecule that's essential for DHPS to work.
So they're like imposters, tricking the enzyme.
Exactly. They sneak into the octave site of DHPS, where PABA usually binds, but they block the enzyme's activity.
Like a fake key. It fits but won't turn.
Perfect analogy. This blockage prevents DHPS from making dihydrofolic acid, a key ingredient in the folic acid pathway.
So they disrupt the production line at a crucial step. What happens next?
Without dihydrofolic acid, bacteria can't make tetrahydrofolic acid.
Tetrahydrofolic acid.
Yeah, that's the active form of folic acid. They need it for DNA replication, RNA synthesis, and making amino acids.
So one blocked step leads to a whole cascade of disruptions.
Exactly. And because we get folic acid from our diet, sulfonamides only target bacteria, not our own cells.
Targeted strike. That's some smart pharmaceutical warfare.
Indeed. It all comes down to understanding the differences between bacterial and human cells.
I'm starting to get it. This is cool. Okay, the next question is about the structure of sulfonamides.
Time to get molecular.
Let's do it.
Yeah.
At the core of any sulfonamide is, of course, the sulfonamide group.
Okay, their defining feature. So what does it look like?
It's a sulfur atom double bonded to two oxygens, single bonded to a nitrogen and an aryl or alkyl group.
Okay, so sulfur, oxygen, nitrogen, and a side group.
Right.
This is what gives them their powers.
Precisely. The simplest one is sulfanilamide. It's the OG sulfonamide. Sulfanilamide. So it's like the prototype, the one that started it all.
You got it. It's got the sulfonamide group attached to a benzene ring with an amino group.
Sulfonamide group, benzene ring, amino group. Is that all there is to it?
Well, most sulfonamides used in medicine are derivatives of sulfanilamide. They've been modified to enhance their properties.
So they took the basic design and made it better.
Exactly. Like sulfamethoxazole has a 5-methylisoxazole group attached.
Sulfamethoxazole, yeah, that's the one often combined with trimethoprim.
Precisely. That combination is incredibly effective.
Right, I remember that from earlier.
Now, another key thing is their similarity to PABA.
Right, the molecule they mimic to trick that DHPS enzyme.
Exactly. Both have a benzene ring and an amino group in similar positions.
Okay, so that's what makes them such good imposters. What's the difference between them, though?
The big difference is that sulfonamides have the sulfonamide group, while PABA has a carboxylic acid group.
So that tiny difference is what allows them to bind to DHPS, but stops the enzyme from working. Amazing how small changes can make such a big difference.
It's all about the molecular structure.
This is like molecular detective work. All right, let's switch gears to quinolones. Explain the mechanism of action of quinolones. They're the ones that disrupt DNA, right?
Right. Remember DNA gyrase and topoisomerase Phi?
Yeah, the DNA organizers.
Quinolones target those enzymes.
We talked about that before, but how do they actually do it?
They form a stable complex with the enzyme and the DNA.
A three-way handshake.
Exactly. This complex stops the enzyme from doing its job.
So they're stuck, can't manage the DNA properly.
Right. Normally, these enzymes cut and reseal DNA strands. It's all very controlled.
Right, like a delicate dance.
But when quinolones are bound, the enzymes can't reseal the breaks.
The scissors get stuck.
Exactly. This causes double-strand DNA breaks, which are very dangerous for bacteria.
It's like cutting their DNA, causing serious damage.
And it gets worse. The quinolone enzyme DNA complex also interferes with DNA repair.
So the repair crew can't fix the damage.
Exactly. This leads to bacterial cell death. Now, quinolones are selective for bacteria.
We talked about that earlier. How do they know to target bacterial cells specifically?
They have a higher affinity for bacterial DNA gyrase and topoisomerase 5e compared to human versions of these enzymes.
So they preferentially bind to the bacterial enzyme.
Well, exactly. It's all about those structural differences between bacterial and human enzymes.
It's fascinating how these tiny differences can be exploited. It's like they have a secret code to attack only the bad guys.
It is remarkable. And just like with sulfonamides, we need to be mindful of bacterial resistance.
Right.
Overuse and misuse of quinolones can lead to resistant bacteria.
It's that ongoing arms race we talked about.
Exactly. We always have to be careful and use antibiotics responsibly.
Okay, message received.
Ugh.
So where do we go from here? What else is there to learn about these fascinating drugs?
Well, let's dive deeper into their applications and see how they're used in the real world to fight infections. Ready for the next round?
Absolutely. Bring on the next question.
All right. This one asks about the applications of sulfonamides. So let's see how these molecular spies are deployed in the real world.
I'm ready to see them in action. I know they've been around for a while, so I'm curious about how relevant they still are today.
Well, sulfonamides have been around for decades, and while newer antibiotics have emerged, they're still valuable in certain situations.
So they're not the frontline soldiers they once were, but they still have their place.
Exactly. They're still effective against certain bacteria, have a good safety profile, and have unique properties that make them useful in specific clinical scenarios.
Okay, I'm intrigued.
Yep.
What are some of those scenarios where sulfonamides are still a go-to choice?
One of their main uses is for urinary tract infections, or UTIs,
UTIs, those are so common.
especially in women.
Right, and I've heard that sulfonamides are often used for UTIs.
Yes, especially cotrimoxazole, the combination of sulfamethoxazole and trimethoprim. It's often the first choice for uncomplicated UTIs.
So for those straightforward UTIs, sulfonamides are often the best option.
They're effective against the common bacteria that cause UTIs like E. coli.
Okay, so they cover the usual suspects. What makes them particularly well suited for UTIs?
One reason is that sulfonamides concentrate in the urine, reaching high levels in the urinary tract.
So they're delivered right to the site of infection.
Exactly. But remember, bacterial resistance to sulfonamides has increased in some areas.
Right, that pesky resistance always seems to pop up.
So while they're still used for UTIs, other antibiotics might be preferred in areas with high resistance rates.
Got it. So it's a decision based on the specific bacteria and local resistance patterns. What other infections are sulfonamides used for?
Another important use is for pneumocystis pneumonia, or PCP.
Pneumocystis pneumonia? That sounds pretty serious.
It is. It's an opportunistic infection that mainly affects people with weakened immune systems, like those with HIV/AIDS.
So it's a threat when the immune system is compromised.
Exactly. Cotrimoxazole is used to both treat and prevent PCP in these individuals.
So it's a vital tool for protecting vulnerable people.
They're also used for bronchitis, sinusitis,
Okay.
otitis media, skin infections, even burns. Topical sulfonamides like silver sulfatazine cream are used to prevent infections in burns.
Wow, that's a pretty wide range of applications. They're more versatile than I thought.
Exactly. And they're also used for some less common infections. For instance, nocardiosis.
Nocardiosis, what's that?
It's an infection caused by nocardia bacteria. It can affect the lungs, skin, and even the brain.
Sounds serious.
It can be. And sulfonamides are the go-to treatment. They're also used for toxoplasmosis.
Toxoplasmosis, another one I'm not familiar with.
It's a parasitic infection. It can be really dangerous during pregnancy and for people with weakened immune systems.
So they're even effective against parasites. That's impressive.
And in some parts of the world, they're still used for malaria.
Really?
Well,
Malaria?
yep.
Wow. So they have a role in fighting this global health problem, too.
Now, besides being antibacterial agents, some sulfonamide derivatives have other uses.
Oh. They can do more than just fight bacteria.
Yep. Some are used as diuretics to treat high blood pressure and edema.
So they help regulate fluid balance.
Exactly. Others are used as anti-diabetic agents to lower blood sugar levels in type 2 diabetes.
Wow, that's amazing. They're like multi-purpose tools.
They really are. They play important roles in many areas of medicine.
Well, I'm definitely feeling a lot more knowledgeable about sulfonamides now.
Good.
But we're not done yet. What about those quinolones, the other DNA disruptors? I want to learn more about their applications, too.
Let's do it. Quinolones are real workhorses in the clinic. They have broad spectrum activity and excellent tissue penetration, making them effective against a wide range of bacterial infections.
So they're like those special forces units that can handle almost any situation.
Precisely. They're particularly useful against gram-negative bacteria, including those that are resistant to other antibiotics.
So they're called in when the first-line defenses have failed.
Sometimes, yes. They're a valuable option when dealing with tougher, more resistant bacteria.
Okay, so where are these quinolones deployed? What kind of infections do they fight best?
Well, one of their main targets is the respiratory system. They're very effective against a wide range of respiratory tract infections.
Respiratory tract infections. Those are so common, especially during cold and flu season.
Exactly. Quinolones are great for treating pneumonia, bronchitis,
Okay.
even hospital-acquired pneumonia.
Wow, so they cover a lot of ground in the respiratory system, both in the community and in the hospital. What makes them so effective in these cases?
Well, they're active against many common respiratory pathogens, including Streptococcus pneumonia, Haemophilus influenza,
Okay.
even those atypical bacteria like Mycoplasma pneumonia.
So they're like a broad-spectrum weapon that can handle all those different bacteria.
Precisely. And they have excellent tissue penetration, so they reach high concentrations in the lungs.
It's like delivering those troops right to the front line. Now, we talked about sulfonamides being used for UTIs. Do quinolones play a role there, too?
They do. Quinolones are effective against a bunch of urinary tract pathogens.
Okay.
E. coli, Klebsiella pneumonia, and even Pseudomonas aeruginosa.
Pseudomonas aeruginosa. Isn't that one of those superbugs?
You're right. It's a tough one, but quinolones are often effective against it, especially in UTIs. They're often reserved for complicated UTIs, like those involving the kidneys or prostate, or when there's resistance to other antibiotics.
So they're like the heavy artillery.
You could say that.
I like that analogy.
Huh.
So besides respiratory and urinary tract infections, what else are quinolones used for?
Well, they're also used for intra-abdominal infections.
Intra-abdominal infections. What are those?
Infections within the abdominal cavity, they can be serious, especially if they involve the peritoneum.
The peritoneum.
Yeah, the lining of the abdominal cavity.
Okay.
Quinolones are often used for these infections because they penetrate well into abdominal tissues.
So getting those troops to the right location.
Exactly. They're also used for skin infections, bone infections, joint infections,
Wow.
even some sexually transmitted infections, though their use for STIs has decreased because of resistance.
Okay, so they're really versatile.
They are. They even have some unique applications. For example, ciprofloxacin is used for malignant otitis externa.
Malignant otitis externa. What's that?
It's a severe ear infection that can spread to the skull. It mostly affects people with diabetes or weakened immune systems, and it's often caused by Pseudomonas aeruginosa.
Ah, that tough bacterium again. So ciprofloxacin is key in fighting that infection.
Yes, it is. It's also used to treat and prevent anthrax.
Wow, anthrax. So quinolones even play a role in bioterrorism preparedness.
They do. They're also effective against plague.
So they're like those elite soldiers trained to handle the most dangerous missions. This is incredible, but I know there's a downside too. We talked about resistance, and I remember you mentioning side effects earlier.
Yes, quinolones are not without risks.
Okay, so let's dive into that. This next question asks, describe the adverse effects of quinolones.
All right, so even the most effective drugs can have unwanted effects, and quinolones are no exception. We need to be aware of their potential side effects.
Right, it's all about weighing the benefits and the risks.
Exactly. Now, one of the most concerning side effects is tendinopathy.
We talked about that earlier. It can weaken tendons, right? Especially the Achilles tendon.
Exactly. And this can happen even in people with no history of tendon problems. The risk is higher in older adults, those taking corticosteroids, and those with kidney problems.
Okay, so those are all red flags to watch out for. What are the symptoms?
They can range from tendon pain and swelling to a complete tendon rupture.
Or rupture.
Yeah, a complete tear. That's a serious injury that often needs surgery.
Wow, definitely not something to ignore. What about other effects on muscles and joints?
Well, joint pain and muscle pain are fairly common side effects of quinolones.
So aches and pains.
Yeah.
Okay.
Usually mild and temporary, but they can be bothersome for some people.
Yeah, okay, good to know. Can quinolones affect the brain and nerves?
Yes, they can. They can cross the blood-brain barrier, meaning they can get into the brain.
So they can directly affect the brain. That's kind of scary. What kind of effects can they have?
Well, they can cause all sorts of neurological side effects.
Okay.
Headaches, dizziness, insomnia, anxiety, and even agitation. And in more severe cases, they can cause confusion, delirium, seizures, even psychotic reactions.
Wow, so they can really mess with your head. Are some people more likely to experience these effects than others?
Yeah, older adults and people with existing neurological conditions are definitely at higher risk.
So extra caution is needed in those groups. What about effects on the nerves outside the brain?
They can cause peripheral neuropathy.
Peripheral neuropathy, what's that?
It's nerve damage that can cause numbness, tingling, burning pain, weakness.
Where does that happen?
Usually in the hands and feet.
That sounds awful.
It can be, and in some cases, the damage is permanent.
So another serious risk to consider. What about the heart? Can quinolones affect the cardiovascular system?
They can. One of the most worrying effects is QT prolongation.
QT prolongation. I've heard that term, but what does it actually mean?
It's basically an abnormality in the heart's electrical activity.
Okay.
It can increase the risk of a heart rhythm problem called torsades de pointe.
Then that's serious, right?
Very serious. It can be life-threatening. Quinolones block certain potassium channels in the heart.
Okay.
That's what can lead to QT prolongation.
Gotcha. Is anyone more at risk for this?
Yeah, people with pre-existing heart conditions, electrolyte imbalances, or people taking other QT prolonging drugs.
So careful monitoring is essential, especially for those at higher risk. Any other heart-related effects to be aware of?
Yes, some studies suggest a possible link between fluoroquinolones and a higher risk of aortic aneurysm and dissection.
Aortic aneurysm, that's when there's a bulge in the aorta, right?
Yes, the main artery that carries blood from the heart. And a dissection is a tear in the aorta's inner layer. It can be life-threatening.
So those are both very serious conditions. How high is the risk with quinolones?
The risk is thought to be small, but it's definitely something to consider when weighing the risks and benefits.
Okay, another factor to think about. What about stomach upset? Can quinolones cause gastrointestinal issues?
Oh, yeah. Nausea, vomiting, diarrhea, abdominal pain. Those are all common side effects.
Okay, so those are pretty standard side effects for many drugs. Anything more serious we should know about?
One concern is their potential to cause C. diff infection.
C. diff, that's the one that causes severe diarrhea, right?
Exactly. Quinolones, like a lot of broad-spectrum antibiotics, can upset the balance of bacteria in the gut.
Oh?
That can allow C. diff to grow too much.
So they can actually create an environment where these harmful bacteria can thrive.
Exactly. Now, on top of that, quinolones can also affect blood sugar levels. They can cause both low blood sugar, hypoglycemia, and high blood sugar, hyperglycemia.
Wait, they can cause both.
Yeah.
How is that possible?
We don't fully understand how it works, but it seems they can affect insulin secretion and sensitivity.
So they mess with how the body regulates blood sugar.
Precisely. And these blood sugar fluctuations can be especially risky for people with diabetes.
Definitely something to watch out for. What about allergic reactions?
Those can happen too. Rashes, itching, swelling. In more severe cases, angioedema.
Angioedema.
That's swelling of the face, lips, tongue.
Okay.
Or even anaphylaxis, which is a life-threatening allergic reaction.
Does sound pretty scary.
They can be, and those reactions definitely need immediate medical attention. And there are even more side effects, though they're less common. They can affect the liver, the kidneys, blood cells,
Wow.
even the eyes and ears.
It seems like quinolones can affect almost every part of the body.
It's important to remember that side effects don't happen to everyone.
Right.
Many people tolerate these drugs well, but because of the potential for serious side effects, we have to be very careful.
It's about weighing the benefits against the risks.
Exactly. Choosing the right patients, the right doses, and watching out for any problems.
Well, that was a pretty intense look at the potential downsides of quinolones. Powerful drugs, but they come with responsibilities.
Absolutely. We have to use them wisely.
All right, ready for the next question?
Get me.
Okay, this one asks, describe the various classes generations of quinolones. So let's zoom out a bit and see how these DNA disruptors have evolved.
Sure. Like most drugs, quinolones have gone through several generations of development.
Makes sense. Always trying to improve, right?
Right. Each generation builds on the last, trying to be more effective and overcome limitations.
Okay, I'm ready for a history lesson in quinolone evolution. Where do we start?
Let's go back to the 1960s, the first generation quinolones.
The pioneers. What were they like?
They were effective against some gram-negative bacteria, especially those causing UTIs, but they had their limits.
So they were the first troops on the ground, but they weren't as advanced as later generations.
A good way to put it, they had a narrow spectrum of activity,
Okay.
weren't as potent, and they didn't distribute well in the body.
So like those early rifles, effective for some things, but not very powerful or versatile.
Exactly. The best example is nalidixic acid.
Nalidixic acid, I think I've heard of that one. So it was mainly used for uncomplicated UTIs.
Right.
Okay. And then what happened?
Then came the second generation quinolones in the late 70s and 80s. Big improvements.
Okay, so what kind of upgrades did they get?
They had a broader spectrum of activity,
Okay.
meaning they were effective against a wider range of bacteria, including some gram-positives.
So it's like upgrading from a rifle to a machine gun.
I like that.
Hmm.
They were also more potent and had better pharmacokinetic properties.
Hmm.
That means they were absorbed better, reached higher concentrations in the body, and lasted longer.
So they were like high-tech weapons, more precise, powerful, and longer lasting.
Exactly. One key change was adding a fluorine atom to the quinolone ring.
Fluorine.
Yep.
Oh.
This is called fluorination, and it really boosts their potency and activity.
So it's like adding a special coating that makes them more lethal.
You got it. Most quinolones we use today are fluoroquinolones.
Okay, so the second generation was the rise of the fluoroquinolones.
Mm-hmm.
What are some of the big names from this generation?
Oh, some of the most famous antibiotics out there.
Like what?
Ciprofloxacin, norfloxacin, afloxacin, enoxacin.
Ciprofloxacin, afloxacin, those are definitely failure. They're like the rock stars of the antibiotic world.
They are very important drugs used all over the world.
Okay, what about the third generation quinolones?
They came along in the 1990s. They had even better activity against gram-positives, especially Streptococcus pneumonia.
The one that causes pneumonia.
Right.
Yeah.
They were designed to be more effective against respiratory pathogens. That's why we call them respiratory fluoroquinolones.
So they're like specialized units trained for combat in the respiratory system.
Exactly. Levofloxacin and gemifloxacin are good examples.
Levofloxacin, another one I've heard of. It's amazing how each generation builds on the previous one, making these drugs more powerful and versatile.
It really is. And then we have the fourth generation, the newest kids on the block.
Okay, tell me about them. The latest and greatest in quinolone evolution.
These have the broadest spectrum of activity. They cover gram-negatives, gram-positives, even anaerobic bacteria.
They're like those all-terrain vehicles that can handle any kind of road.
Exactly. They're incredibly versatile. The key players are moxifloxacin and delafloxacin.
More new names. I'm going to need flashcards to keep track of all these quinolones.
It is a lot to remember. But remember, even with all these advancements, we still have to worry about side effects and resistance.
Right, those challenges never really go away.
We have to use them carefully and appropriately.
Absolutely. Well, that was an incredible journey through quinolone history. It's amazing to see how these drugs have been refined and improved over time.
It's a great example of how science is constantly working to find better treatments.
Now, we did touch on some side effects earlier, but I think it deserves more attention. This question asks, describe the adverse effects of quinolones. Let's really unpack those potential downsides.
Good idea. It's crucial to understand both the benefits and the risks of these drugs.
Right. It's all about making informed decisions. So, what are some of the things we should be aware of?
Well, we already talked about tendinopathy and the risk of tendon rupture. That's definitely one of the most serious concerns.
Right. It can affect even healthy people, and the risk is higher in certain groups.
Exactly. Older adults, people on corticosteroids, and those with kidney problems are more vulnerable.
So those are red flags to watch for. And what about those neurological effects? They can affect the brain, right?
They can. They can cause headaches, dizziness, insomnia, even anxiety.
Those are pretty common side effects of a lot of medications. Anything more serious to worry about?
In some cases, yes.
Okay.
They can cause confusion, delirium, even seizures. And in rare cases, they've been linked to hallucinations and other psychotic-like symptoms.
Wow, that's pretty intense. It seems like those effects are more likely in older adults or those with pre-existing neurological conditions.
You're right. We have to be especially careful in those groups.
Okay. So tendon problems, brain and nerve issues. Anything else?
Well, what about the heart?
Oh, right. We touched on those cardiovascular effects. QT prolongation, aortic aneurysm, and dissection.
Exactly. Those are serious potential complications, especially QT prolongation. That can lead to life-threatening heart rhythm problems.
So careful monitoring is essential, especially for patients with heart problems or those taking other medications that can affect heart rhythm.
Absolutely. And then there's the gastrointestinal system.
Stomach upset, I know we talked about that. Nausea, vomiting, diarrhea. But is there anything more serious than just an upset stomach?
Yes. Quinolones can increase the risk of C. diff infection. That's a severe bacterial infection that can cause debilitating diarrhea.
Right, we talked about how they can disrupt the balance of bacteria in the gut.
Exactly. And then there's the effect on blood sugar. Quinolones can cause both high and low blood sugar, which can be dangerous for people with diabetes.
So careful monitoring is crucial for them as well.
And of course, there's always the risk of allergic reactions.
Like rashes, itching, swelling.
Eh.
The usual suspects. But you mentioned some more serious allergic reactions too.
Right. Angioedema, which is swelling of the face and tongue, and anaphylaxis, which is a life-threatening allergic reaction.
So those are definitely emergencies that need immediate medical attention.
Absolutely. And while less common, quinolones can also affect the liver, kidneys, blood cells, even the eyes and ears.
Wow. So quinolones are powerful drugs, but they come with a long list of potential side effects.
That's why they should only be used when clearly necessary and with careful consideration of the risks and benefits for each patient.
Okay, so what else is there to know about quinolones? What haven't we covered yet?
Well, we talked about the different generations, but we haven't really explored those in detail.
Right, the different generations of quinolones. I'm curious to learn more about that.
It's like a family tree. You have the first generation, the pioneers, then each generation gets a bit more sophisticated.
So it's a story of constant evolution, with each generation building on the successes of the previous one.
Exactly.
I'm ready to dive into that family tree. What are the key differences between these generations?
Let's start with the first generation. We already mentioned nalidixic acid.
Right, the granddaddy of them all, primarily used for UTIs, right?
Exactly. It had a narrow spectrum of activity, wasn't very potent, and didn't distribute well in the body.
So effective for some things, but limited in its power and reach.
Right. Then came the second generation, which brought some big improvements.
Like what?
Well, they had a broader spectrum of activity. They were effective against more types of bacteria, including some gram-positives.
So more versatile.
Exactly. They were also more potent and had better pharmacokinetics. That means they were absorbed better, reached higher concentrations in the body, and lasted longer.
So they were like those upgraded weapon systems, more precise, powerful, and longer lasting.
A great analogy. The key innovation in this generation was the addition of a fluorine atom to the quinolone ring. We call that fluorination.
Fluorination, I remember you mentioned that earlier. It enhances their potency and activity.
Exactly. And most of the quinolones we use today are fluoroquinolones. They have that fluorine atom.
Okay, so the second generation marked the rise of the fluoroquinolones. What are some of the big names from this generation?
Oh, some of the most familiar ones. Ciprofloxacin, norfloxacin, afloxacin, enoxacin. These are all second-generation fluoroquinolones.
Ciprofloxacin and afloxacin. Those are household names in the world of antibiotics. They're used for so many different infections.
They are. And then came the third-generation quinolones in the 1990s.
Okay, what improvements did they bring to the table?
They had even better activity against gram-positives, particularly Streptococcus pneumonia, the one that causes pneumonia.
So they were even more effective against those respiratory pathogens.
Exactly. They were specifically designed to target those bugs. That's why we call them respiratory fluoroquinolones.
Like a special forces unit trained for combat in the lungs.
Exactly. Levofloxacin and gemifloxacin are two good examples.
More familiar names. It's amazing to see how each generation builds on the previous one, making them more and more effective.
It really is. And finally, we have the fourth generation, the most recent additions to the quinolone family.
The cutting edge. What sets them apart?
Well, they have the broadest spectrum of activity of all. They're effective against a wide range of gram-negative, gram-positive, and even anaerobic bacteria.
So they're like those all-terrain vehicles that can handle any kind of road.
Exactly. They're incredibly versatile, which makes them valuable for treating a wide variety of infections.
So who are the stars of this generation?
Moxifloxacin and delafloxacin are the key players here. Moxifloxacin is considered the broadest spectrum quinolone of all, with excellent activity against many different types of bacteria.
Moxifloxacin. I'm starting to feel like I need a cheat sheet to keep track of all these quinolones.
It can definitely be a lot to take in, but the key takeaway is that each generation builds on the successes of the previous one.
Right. Always striving for improvement. But even with all these advancements, we can't forget about resistance and those potential side effects.
Absolutely. We always need to be mindful of those challenges and use these drugs responsibly.
Okay, I'm ready for the next question. What other fascinating insights do we have in store?
Well, let's shift gears a bit and explore a different class of antimicrobial agents.
Okay, I'm intrigued. What's next on our agenda?
This question asks about polymyxins. Specifically, it wants us to explain the mechanism of action and uses of polymyxins.
Polymyxins. Those are the ones that disrupt the bacterial cell membrane, right?
You got it. They're often used as a last resort for multi-drug resistant bacteria.
So they're like the big guns we bring out when the other weapons have failed.
You could say that. They're powerful but also potentially toxic, so they have to be used carefully.
Okay, let's break it down. How do they actually work? What's their mechanism of action?
Well, polymyxins are cationic peptides. That means they have a positive charge.
Okay.
And that positive charge allows them to interact with the bacterial cell membrane, which has a negative charge.
So it's like magnets, opposites attract.
Exactly. Now, gram-negative bacteria have an outer membrane that's rich in lipopolysaccharide, or LPS.
LPS, that's a toxin that can cause serious problems during infections, right?
You're right. It's a major player in the inflammatory response triggered by gram-negative infections.
Okay. So where do the polymyxins come into play?
Polymyxins have a high affinity for LPS. They bind to it and displace the calcium and magnesium ions that normally stabilize the outer membrane.
So they disrupt the structure of that protective outer membrane.
Exactly. This disruption creates pores or channels in the membrane, making it leaky.
So it's like punching holes in their armor.
Exactly. Essential cellular components start leaking out, and the bacteria can't maintain their internal environment.
And that leads to cell death.
Precisely. Now, polymyxins are primarily effective against gram-negative bacteria because of their strong interaction with LPS.
Okay, so they're like specialized weapons specifically designed for gram-negative foes.
Right. They're not very effective against gram-positive bacteria because they lack that LPS-rich outer membrane.
Got it. So what are the main clinical uses of polymyxins? You mentioned they're often used as a last resort.
You're right. They're usually reserved for infections caused by multi-drug resistant gram-negative bacteria, those superbugs that are resistant to most other antibiotics.
So they're like the elite forces we deploy when the situation is really dire.
Exactly. They can be lifesavers in those cases.
What kind of infections are we talking about specifically?
Well, they're commonly used for complicated urinary tract infections, bloodstream infections, pneumonia, and even meningitis caused by these resistant bacteria.
Okay, so they're used for serious life-threatening infections when other options have failed.
Precisely. Now, polymyxins are available in both intravenous and topical formulations.
Topical formulations, so they can be applied directly to the skin.
Exactly. Polymyxin B is often combined with other antibiotics in topical ointments or creams to treat skin infections.
Okay, so powerful drugs, but with limited uses due to their potential toxicity. We talked about that earlier, but can you remind me what kind of side effects they can cause?
Sure. The main concerns with polymyxins are nephrotoxicity and neurotoxicity.
Nephrotoxicity. That means they can damage the kidneys.
Exactly. And neurotoxicity means they can damage the nerves.
Okay, so that's pretty serious.
It is. These side effects are more common with intravenous administration and higher doses.
So careful monitoring is crucial.
Absolutely. Especially kidney function tests.
Well, that's a lot to consider. Polymyxins are definitely not drugs to be taken lightly. But it's reassuring to know that we have these powerful weapons in our arsenal when those superbugs come knocking.
They are valuable tools, but as with all antibiotics, we have to use them responsibly and judiciously.
Okay, what else is there to learn about these microbial warriors? What's next on our agenda?
Let's tackle this question. Explain the mechanism of action of antifungal drugs. So we're shifting our focus from bacteria to fungi.
Okay, I'm ready to explore the world of fungi fighting strategies.
Remember how we talked about fungi being eukaryotic organisms like us?
Right, they have cells that are more similar to ours than bacteria, which makes them harder to target selectively.
Exactly. But there are still some key differences that we can exploit.
So finding those chinks in their armor that our own cells don't have.
Precisely. Now let's talk about some of the main mechanisms by which antifungal drugs work.
Okay, I'm ready for a lesson in fungal warfare.
One of the most common mechanisms is targeting ergosterol. Ergosterol. It's a sterol, a type of lipid,
Okay.
and it's a crucial component of fungal cell membranes, similar to cholesterol in our cell membranes.
So it's like the cholesterol of fungi.
Exactly. Now, many antifungal drugs target ergosterol in different ways.
Okay, so how do they do that? What are some examples?
One class of antifungal drugs is called the polyenes.
Polyenes. I'm getting flashbacks to cell membranes and punching holes.
You're on the right track. Polyenes, like amphotericin B and nystatin, bind directly to ergosterol in the fungal cell membrane.
Okay, so they're like those membrane disruptors we talked about earlier, but specifically targeting fungal cells.
Exactly. They bind to ergosterol and form pores or channels in the membrane, making it leaky.
So same concept as polymyxins, but with a different target.
Precisely. Those leaky membranes can't maintain the cell's internal environment, leading to fungal cell death.
Okay, so polyenes are like those membrane-disrupting missiles, but specifically aimed at fungi. What other ergosterol targeting strategies are there?
Another class of antifungal drugs is called the azoles.
Azoles. I think I've heard of those. Aren't they pretty commonly used?
They are. They're often the first-line treatment for many fungal infections.
Okay, so they must be pretty effective. How do they target ergosterol?
Azoles, like fluconazole, itraconazole, and voriconazole, inhibit an enzyme called 14-alpha demethylase.
14-alpha demethylase. That's a mouthful. What does this enzyme do?
It's a key player in ergosterol synthesis. It catalyzes a crucial step in the pathway that produces ergosterol.
So by inhibiting this enzyme, azoles essentially block the production of ergosterol.
Exactly. They disrupt the fungal cell's ability to make this essential component of its membrane.
And that weakens the membrane, making the fungal cell vulnerable.
Precisely. Now, besides those ergosterol targeting strategies, there are other mechanisms that antifungal drugs employ.
Okay, I'm ready to hear about those. What other fungal vulnerabilities do we exploit?
Well, another target is the fungal cell wall.
The cell wall, just like with bacteria.
Exactly. Remember, the fungal cell wall is different from the bacterial cell wall.
Right, they have different components.
Precisely. And we can target those differences. One important class of antifungal drugs that targets the cell wall is the echinocandins.
Echinocandins, never heard of those. How do they work?
Echinocandins, like caspofungin, mycofungin, and inidulafungin, inhibit an enzyme called 1-3-beta-D-glucan synthase.
Another long enzyme name. What does this one do?
It's essential for the synthesis of beta-glucan, a major component of the fungal cell wall.
So it's like disrupting the construction of their protective wall.
Exactly. By inhibiting this enzyme, echinocandins prevent the formation of beta-glucan, weakening the cell wall and making the fungal cell more susceptible to bursting.
So it's a similar concept to how some antibiotics target the bacterial cell wall, but with a different enzyme and a different component of the cell wall.
Exactly. Now, besides those main mechanisms, there are other less common targets for antifungal drugs.
Okay, what are those?
Some antifungal drugs, like flucytosine, interfere with fungal DNA and RNA synthesis.
Right.
Others, like griseofulvin, disrupt microtubule function, which is important for cell division. And some, like cyclopirox, have multiple mechanisms of action that we don't fully understand yet.
So there's still a lot we don't know about how some of these antifungal drugs work.
That's right.
But the bottom line is that we have a variety of weapons in our arsenal to combat fungal infections. It's all about choosing the right drug based on the specific type of fungus, the site of infection, and the patient's overall health.
Precisely. Now, just like with antibiotics, antifungal resistance is a growing concern.
Right. Those pesky microbes are always finding ways to outsmart us.
Exactly. So we have to use these drugs responsibly to minimize the development of resistance.
Okay, message received. So what other microbial mysteries do we have to unravel?
Well, let's dive into the world of antiviral agents.
Okay. Those are the ones that target viruses, right? Those tricky little invaders that aren't even cells.
Precisely. And that makes them particularly challenging to target.
Right, because they hijack our own cells to replicate. So how do we fight back against them?
Well, the key is to target specific steps in the viral replication cycle without harming our own cells.
So we need to find those points of vulnerability in their life cycle where we can disrupt their plans.
Exactly. Now, let's break down those different stages and see how antiviral drugs intervene.
Okay, I'm ready for a lesson in viral warfare tactics.
The first stage is viral attachment and entry.
So that's how the virus actually gets into our cells.
Right. Viruses have specific proteins on their surface that bind to receptors on our cells. It's like a lock and key mechanism.
Okay.
Now, some antiviral drugs block this attachment process.
So they're like those security guards that prevent unauthorized entry.
Exactly. For example, the drug Marevirac, which is used to treat HIV, blocks the CCR5 receptor on immune cells, preventing HIV from binding and entering those cells.
Okay, so it's like changing the locks on the doors so the virus can't get in. What happens once the virus does manage to get inside?
Well, the next stage is viral uncoating.
Uncoating, what's that?
Once inside the cell, the virus needs to release its genetic material, either DNA or RNA, so it can take over the cell's machinery.
Okay, so it's like shedding its coat to reveal its true identity.
Exactly. Now, some antiviral drugs interfere with this uncoating process. For example, amantadine and ramantadine, which are used to treat influenza A, prevent the virus from uncoating and releasing its RNA.
So it's like trapping the virus inside its own coat, preventing it from doing any damage.
Precisely. Now, once the virus has released its genetic material, the next stage is nucleic acid synthesis.
That's when the virus starts making copies of its DNA or RNA, right?
Exactly. And this is a prime target for antiviral drugs. There are many different drugs that interfere with this process, targeting different enzymes involved in viral DNA or RNA replication.
So it's like disrupting their printing press, preventing them from making more copies of themselves.
Exactly. For example, acyclovir, which is used to treat herpes simplex virus and varicella zoster virus, is a nucleoside analog.
A nucleoside analog, what's that?
It's a molecule that looks a lot like the building blocks of DNA, the nucleosides.
Okay.
Acyclovir gets incorporated into the viral DNA chain during replication, but it acts as a chain terminator.
So it's like a saboteur that sneaks into the production line and messes everything up.
Precisely. This stops the viral DNA from being copied properly, blocking viral replication.
Okay, that's pretty clever. So we've got drugs that block attachment, drugs that prevent uncoating, and drugs that interfere with nucleic acid synthesis. What happens next in the viral life cycle?
Well, once the virus has made copies of its genetic material, it needs to use that material to produce viral proteins.
Right. Proteins are the workhorses of the cell, so the virus needs them to do its dirty work.
Exactly. And this stage, viral protein synthesis, is another target for antiviral drugs.
So it's like attacking their factories where they make those essential proteins.
Precisely. Now, there are several ways to disrupt viral protein synthesis. One way is to target the ribosomes, just like some antibiotics do in bacteria.
Okay.
But remember, viral ribosomes are different from human ribosomes, so we can target them selectively.
So we need to find those subtle differences to avoid harming our own cells.
Exactly. For example, some antiviral drugs like plecanaril, which is used to treat certain enterovirus infections, bind to viral capsid proteins, preventing the assembly of new viral particles.
Capsid proteins.
Yeah, those are the proteins that make up the outer shell of the virus.
Okay, so it's like interfering with the construction of their protective armor.
Precisely. And finally, once the virus has made all its components, it needs to package them up into new viral particles and release them to infect other cells.
Right, so it's like their grand finale, their exit strategy.
Exactly. And this stage, viral assembly and release, is another target for antiviral drugs. Host. Some antiviral drugs like oseltamivir and zanamivir, which are used to treat influenza, inhibit neuraminidase, an enzyme that's essential for the release of new viral particles from infected cells.
So they're like those border patrol agents that prevent the virus from escaping and spreading to other cells.
Exactly. Now remember, developing antiviral drugs is particularly challenging because viruses are so reliant on our own cellular machinery.
Right, they're like those sneaky spies that blend in with the crowd.
Exactly. So we have to be very clever in designing drugs that selectively target viral processes without harming our own cells.
It's a delicate balancing act.
Yeah,
But it's incredible how scientists have been able to develop so many effective antiviral drugs despite these challenges.
it's a testament to the ingenuity and perseverance of researchers in the field of antiviral therapy.
Well, I feel like we've covered a tremendous amount of ground in this deep dive, from those sneaky sulfonamides to those powerful quinolones, those last resort polymyxins, and those targeted antifungal and antiviral agents. It's been an incredible journey through the world of microbial warfare.
And remember, listener, understanding how these drugs work isn't just about acing your exams. It's about appreciating the remarkable advances in science that are helping us combat these microscopic foes that can impact human health.
Exactly. It's about empowering yourself with knowledge and understanding the intricate ways in which we're fighting back against these tiny but mighty invaders. But before we sign off, I have one last burning question. We've talked about resistance to antibiotics and antifungal drugs. What about antiviral resistance? Is that a thing?
It's definitely a thing, and it's a growing concern in the field of antiviral therapy.
Okay, so those viruses are just as crafty as bacteria and fungi when it comes to evolving and developing resistance.
Unfortunately, yes. Viruses can mutate rapidly, and those mutations can sometimes confer resistance to antiviral drugs.
So it's like a constant arms race, always trying to stay one step ahead of those evolving viruses.
Exactly. That's why responsible use of antiviral drugs, just like with antibiotics and antifungals, is crucial to minimize the emergence of resistance.
Well, that's a sobering thought, but it's important to be aware of these challenges.
It is.
Well, listener, we've reached the end of our deep dive into the world of pharmaceutical biotechnology. We've explored the mechanisms, applications, and challenges of those amazing drugs that are helping us win the war against microbial invaders.
It's been a fascinating journey, and we hope you've enjoyed it as much as we have.
And remember, this is just the beginning of your exploration. The world of pharmaceutical biotechnology is vast and constantly evolving, with new discoveries and challenges emerging all the time. Keep asking questions, keep learning, and keep that thirst for knowledge alive.
Exactly. And good luck with your exams. We're confident that this deep dive has given you the knowledge and understanding you need to succeed.
Best of luck to all of you. Until next time, stay curious and stay informed.